I'm Sophie, this is Anime, and today we'll be talking about gene therapy. And so we'll just kind of give a brief overview of what gene therapy is, and then a little bit of a discussion about the history and recent successes.
So all of us are really familiar with vaccinations, antivirals, antibiotics as cures for infectious disease. And this is a great thing. But there's a large category of diseases that we don't really have a good cure for, including Parkinson's disease, lots of different kinds of cancers, cystic fibrosis, blood diseases, or even immune deficiency. So we have here the severe case of a bubble boy who has severe combined immunodeficiency. All of these categories of diseases are linked in that they affect genes, the genome in some way, and potentially could find a cure through gene therapy.
So, gene therapy, potentially the beginning of an age that will fundamentally change medicine, which sounds kind of sensational, but really this could be what the future of medicine, like advancements in medicine, look like for the next 20 to 50 years. But just to step back a little bit, as a brief overview, gene therapy is the insertion of nucleic acid into a patient's cells in an effort to treat or stop a disease. So generally, you have a cell with a non-functioning gene, you've seen this diagram here, and then take the functioning gene, then you insert the functioning gene into some sort of vector, which will then deliver it to the cell, the target cell, and that will lead to normal function. So, promising method for treating genetic diseases. And there's two main types. So actually, this kind of ties into what we talked about with how there are somatic and germline mutations in class, but there's also somatic and germline types of gene therapy. Somatic being the type where you target somatic cells, but then the therapy isn't necessarily passed on to future generations. And germline being one that targets germline cells, but this typically involves research with stem cells, and it gets into an area that results in it being banned in many countries due to ethical concerns. And there's two main methods of gene therapy. There's in vivo and ex vivo methods. So in vivo being you insert the gene just directly into the patient's cells or the target cells, and the cells uptake the functioning gene. Ex vivo being you take the target cells out, and then you insert the correct gene, and then you insert the cells back into the body. And so in this presentation, we're going to primarily focus on somatic gene therapy, particularly ex vivo methods, as those are the ones we've seen the most advancement in.
So just a really brief history on gene therapy. In 1972, it was kind of first conceptualized by Friedman and Roblin, who wrote an article talking about gene therapy for human genetic disease. And they really saw gene therapy as the future, but before it got really underway, they wanted to make sure they set out ethico-scientific criteria so that gene therapy was used for beneficial methods. So then in 1990, we had the first gene therapy patient, a 4-year-old girl who had adenosine deaminase deficiency, so she was a bubble girl, and it was slightly successful, but she did still have to take the enzyme throughout her life. Then in 2008, we had the first product that completed phase 3 clinical trial, and this was in the EU, where Serapro was an adenovirus vector. And then following that, there's a large chunk of time that was dedicated to really clinical trials, just kind of working through things, continuing the research, and ethical discussions. But then in 2017, it was kind of the big year for gene therapy because it was the year that the FDA approved the first gene therapy treatment. And then in December of the same year, just 1 year ago from today, they approved the first gene therapy for an inherited disease. So kind of a brief overview.
So how does gene therapy actually work? Well, most of the science and the research that's being devoted to it is dedicated to the vector. Otherwise, it's just kind of inserting it into cells and then letting the cells proliferate on their own. But the vector is generally what's required to transport the DNA to the target cells. And so the ideal vector will transfer a specific amount of information to a specific type of cell. And as this chart outlines, there's generally two categories of vectors, viral and non-viral. And Among the non-viral, there can be chemical or physical approaches, chemical being like liposomes or lipoplexes, and physical being methods like electroporation or microinjections, also just the insertion of plasmids and the cells will freely uptake. But generally, these aren't as efficient as the viral vectors. And viral vectors can be split up into two types. There's the integrating and the non-integrating. And an example of the integrating would be like a retrovirus. So they use their RNA as a template to create DNA that's incorporated into the cell genome. So the DNA will be passed on to future cells after the cell divides. And there's also the non-integrating viral vectors. And an example of this are the adenoviruses that introduce double-stranded DNA that's not incorporated into the host genome. So the effects are transient because after division, it won't remain in the cell, but hopefully, ideally, will have some sort of effect on the cell when it's injected first. And a new area of research is on the use of transposons as vectors. And these are kind of, in some ways, seen as the best of both worlds because they're non-viral, so they don't have the potential to elicit a harmful immune response the way viral vectors can. And they're naturally integrating, so they have that effect that isn't transient and will last after cell division. Also, just because they're basically naked DNA, they're easier to produce and they're less expensive to do so. So potentially this is seen as like the new and kind of upcoming exciting field where a lot of research is going into. And we're still at the clinical stage. I mean, I think we just reached the clinical stage. There's like a few trials that are going on with transposon vectors. But there's still a lot of research that's left to be done in this area before we will probably be seeing them making any kind of major changes anytime soon. And an example of a promising transposon that is currently being worked on is the Sleeping Beauty transposon.
So gene therapy overall is just a new field, and so with any new field there come risk. As one article put it, transparency is key to the acceptance of gene therapy. So there are kind of four major problems that have to be ironed out, worked out in order for this to be a viable cure. So you have unwanted immune system reaction. So when the gene is injected into the body could react with inflammation and organ failure. And in 1999, this actually occurred when an 18-year-old took part in a clinical trial and ended up dying 4 days later due to organ failure. Another way that this could potentially go wrong is that it could be targeting the wrong cells, and so you have insertional mutagenesis or oncogenesis. There's also the virus could cause an infection, so it could kind of revert back to its original property and cause that infection, or it could just cause a tumor, which could end up leading to cancer. So none of these are good things, but they're definitely being broached and researched.
And there have been successes, but currently, aside from those two approved methods we talked about in the beginning by the FDA, the only way to receive gene therapy is to participate in a clinical trial, and we have more than 1,800 approved gene therapy trials that have been completed or are still ongoing. I mean, 60% of which are focused on cancer, but a lot is going to come from this. The first unambiguous success of gene therapy was in 2000 when 2 children were successfully cured from severe combined immunodeficiency. And so this was a major milestone, but there was also a sad part to it because other children in the study ended up developing leukemia. So while we did prove that gene therapy, is like an effective method of treatment, the method itself is still not completely risk-free. But then in 2009, there was a gene transfer that halted the progressive brain damage caused by ADL in 2 7-year-old patients. And this time, there weren't any unfortunate side effects like leukemia.
So in 2011, there was a young man that was cured of hemophilia B through an adeno-associated virus vector that delivered factor IX, which is an essential protein for blood clotting. You see his picture in the upper right corner. And he was just one of many in the study that were successful.
And then continuing with the train of success, in 2016, Italian scientists cured 18 children of ADA SCID. So these were children who would have been like the little boy that we saw in the beginning, and now they're completely free and able to live normal lives, and they don't even need to go back for readministration of the enzyme. And in March 2017, a teenage boy in France was cured of sickle cell disease, which, I mean, this is crazy. This is the first time we've ever been able to do anything about this genetic disease, and we've cured it, which, yeah, major milestones are happening.
And then in August 2018, so this was just literally a couple months ago, there was a 3-year-old girl that was cured of spinal muscular atrophy. This disease is generally fatal by the age of 2. And these parents actually lost their first child to this disease. So they enrolled their second daughter in this clinical trial, and they used a viral vector that was able to cross the blood-brain barrier, which is insane. And it entered the central nervous system and cured this little girl. So there's a lot going on in gene therapy, a lot of possible cures for a lot of diseases that up to this point we haven't seen a cure for.
And like we said earlier, this could be what the future of medical development looks like for the next 20 to 50 years. So this is definitely a field to keep your eye on. Thank you.